Unit 8, page 2. What is the central dogma? Let's start out with a warm-up question. What are the three parts of the DNA nucleotide? You may flip back in your notes to look. Pause the video here so you can write your response. Press play when you're ready to check. Let's check the answer to the do now. What are the three parts of the DNA nucleotides? The three parts are the phosphate, the deoxyribose, or sugar, and the nitrogenous base, which is A, T, G, or C. Now, do you remember which one changes and which ones stay the same? Good. The nitrogenous bases are going to change depending on if it's adenine, thymine, cytosine, or guanine. And the phosphate and deoxyribose sugar will make up the backbone of DNA, so they'll remain the same. Now, what is the central dogma of molecular biology? Well, we know that DNA controls our traits and controls who we are, but how does it do this? Well, DNA itself remains the nucleus. It undergoes a process known as transcription to create a messenger known as RNA. RNA will then travel out of the nucleus to the ribosome. At the ribosome, a process called translation will occur where RNA is translated into proteins. Now, these proteins could become receptors, they could become hormones, they could become enzymes, any functional unit needed by the DNA for the cell. So once the gene is activated, this DNA RNA protein stays the same for all living things. So let's get into a little more detail. What is the central dogma? Transcription of DNA to RNA to protein. This dogma forms the backbone of molecular biology and is represented by four major stages. Number 1, DNA replicates its information in a process involving many enzymes in a process known as replication. So this is the DNA copying itself. This is going to happen before cellular division so that each cell gets a copy of the DNA. Number 2, the DNA codes for the production of messenger RNA, otherwise known as mRNA, during the transcription process. Number 3, the mRNA is processed essentially by splicing and cutting. There's actually parts of the DNA code that encode for things that are not necessarily needed to be translated into proteins. So there are these different parts of the mRNA called introns and exons, and certain parts of the mRNA need to be eliminated so that it can actually have a full code. So once this is processed, it migrates or travels from the nucleus to the cytoplasm. In the cytoplasm is where it's going to find the ribosome. So number 4, messenger RNA carries out coded information to the ribosomes. The ribosomes are going to read this information and use it for protein synthesis. This process is called translation. Proteins do not code for the production of a protein, RNA or DNA. They are involved, though, in all biological activity, structural or enzymatic. So again, it's really important to remember the DNA, RNA, protein are what make up your traits. Sometimes we say that genes are being expressed. This means that they are actively making those proteins. So if a gene is being expressed, the DNA actually is going to unwind or unzip so that the mRNA can be made. It's processed, then it leaves nucleus to go to the ribosome and make the correct amino acid or protein. So if a gene is being expressed, that means its protein is being made by that cell. Remember to put down 2 thoughtful questions that you can ask somebody about this video to see whether or not they understood the information.